just using atmospheric oxygen towards tissue culture incubators that are using physiological oxygen. So Randy published in his paper that in the first 500 amino acids there is some NLS activity in Huntington, and it's something that's different than typical known NLSs that go through the carrier-parent data pathway. So Carly Desmond in the lab was working from this and looking at smaller, progressively smaller pieces of Huntington, trying to figure out what was causing the nuclear entry. And at the same time that she had narrowed down a target sequence, Yu-Ming Chuk's lab at UT Southwestern, which was her first paper after leaving Gunther Global's lab in New York, described the consensus structure for what is known as a basic PY-NLS. This is a nuclear localization signal that traffics in and out through the transport pathway, also known as carrier-parent beta 2. It's a completely different pathway of nuclear import. The sequence that Carly was honing in on, turns out, has all the consensus motifs for a PY-NLS. We have the invariant proline tyrosine, an arginine, an appropriate amount of spacing, and then a basic cluster. And this is seen in a large number of proteins in the human proteome, not a huge number, roughly about 120. What's very interesting is almost all of the proteins that contain this type of nuclear localization signal are involved in RNA transport processing from the nucleus, including most of the HNR genes. This signal can function heterologously, so if we have an assay here where we take GFP and beta-galactosidase, we get a nice big protein, and we clone in our test signals. So for example, here's a typical SD40 NLS, and without, we put in the Huntington NLS, and you can see the entry. And importantly, she's been able to describe point mutants that completely knock out this nuclear entry. And it's a simple KKAA mutation in the basic structure of the sequence. So these PY-NLSs are very different than typical NLSs that are used through other pathways. And they're rather unique. What she's showing here is that she can pull down the synthetic peptides transporter that she doesn't see with her mutants. This is a model from Yu-Ming Chuk's papers where she's looking at the fact that, unlike with other NLSs, there are two separated epitopes in a PY-NLS that have an important loop sequence in between. And in the case of Huntington, this loop is unique. All of this is strictly conserved, all the way down to pufferfish and zebrafish. And the loop, at least in the human genome or in the human proteome, is unique to Huntington. This gives us the intriguing possibility that there may be a way to target a small molecule to this loop that would prevent nuclear entry of Huntington. So onward then to figure out what is Huntington doing in the nucleus. And here's Lee's demonstrating proper technique to handle with nitrogen and put in new proposed Canada biosafety guidelines and false hockey pads. So she took cells, and I set her out on a task: stress the cells and let's see what Huntington is doing. But we're going to do this with endogenous Huntington, full length, expressed at endogenous levels. Because once you start stressing cells by overexpression of various fragments of the protein, you're going to see a lot of artifaction effects. So she did this, and she recapitulated some things with overexpression events compared to endogenous immunofluorescence. One of the first effects that she sees is a very fast response, what we call the formation of Huntington heat shock bodies. And they occur on the order of a matter of minutes in response to temperature shock. These shock bodies are transient. They disappear very quickly after the stress is relieved, and they are not P-bodies or stress granules. So if we use markers typical for either stress granules or P-bodies, these are unique structures. But they do contain RNA in the cytoplasm. They're rich in Huntington and they're rich in RNA. And this is immunofluorescence. So the slow stress response, which takes place after about an hour, is the formation of these striking internuclear rods. And honestly, I've never seen anything like this before, so I had her search out the press to see what people have seen this before. We had seen there are nuclear rods that appear in certain diseases, and she did a thorough search, and eventually, with the proper amount of validation, came up with the conclusion that Huntington is in fact localizing to nuclear profilin rods that have been described by James Banberg in Alzheimer's disease. And this was shown with a variety of antibodies to Huntington, both at the amino and the carboxy terminus, saying that this really is full-length protein. striking percentage of the full-length protein that localizes to the rods, and the rods are rich in actin as well, as cofilin rods are. So this is interesting because in Alzheimer's disease, they're really focused on the formation of these rods in the cytoplasm and the potential that they may be affecting vesicular trafficking down in the cytoplasm. However, in the cell lines that we're looking at, we do not see Huntington localizing to the cytoplasmic rods. It only localizes to the nuclear rods, which really no one in Bamberg's lab understands or hasn't had a lot of effort focused towards. So what we can see then is if we compare the rod formation in the wild-type cell lines versus the mutant cell lines, again, full-length Huntington and endogenous levels, we see important differences between wild-type and mutant cells. The rods in mutant cells form and they will disappear. They're very short, but very numerous. Sorry, in wild-type cells. In mutant cells, they're very long, thick, and not very numerous, and they persist. So in experiments where we look at heat shock and then recovery after a few hours and after a day, we can see that we see in the mutant case a situation very similar to what's seen with cytoplasmic rods in Alzheimer's disease, and that's persistence. And in particular, we start seeing actually the formation of aggregates within the nucleus that are rich in cofilin. So she then decided to look at what are the actual protein levels like in these cell lines, and you can see that there's actually an elevated level of cofilin in the mutant cells when normalized against the wild-type cells, about 2.5-fold. The big surprise was that by RT-PCR, the RNA levels are reduced by about a quarter in the mutant cell context. So, just before I go to this, so the next thing that we looked at was knowing that cofilin modulation of actin has important roles in the brain. It also has important roles in hematopoiesis. So this is the work that I presented in California at the beginning of the year, and at the CHDI meeting. And at that point, Sarah Tabrizi came up to us and said, well, you know, maybe you should look at what's going on in patient lymphocytes from the EuroHD study together with Edward Wild. And so I told people to do the Western blot and with cofilin and see if we see increased cofilin levels in these patients. And we didn't. But what we did see was very surprising in that in normal control patients, we see basically one band of free cofilin. But as we look in pre-symptomatic patients, and in particular in advanced stage, clinical stage patients, almost all of the free cofilin has been shifted up into a higher order complex containing cofilin dimers and actin. She's gone on to look at many more patient samples from control, early pre-symptomatic, moderate stages as defined in the clinic, and has seen with the relatively small numbers already statistical power showing that we can track the severity, the clinically described severity of these patients by looking at the ratio of free cofilin to cofilin bound in these active complexes. And she's gone through and done this with several antibodies to cofilin, some of which were monoclonal, some of which were polyclonal. In the end, we've settled on a monoclonal antibody provided to us by James Bamberg because it's the most reproducible. And we're continuing to look at more patients on this. We did longitudinal tracking of 4 patients, and what we're seeing is increased cofilin complex formation in the disease process with 3 out of those 4 patients. The only